Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
When we talk about infectious diseases, few things sound quite as frightening as cerebral malaria. This is where the malaria parasite, Plasmodium falciparum, it doesn't just stay in the blood, it actually starts attacking the brain itself.
And the consequences are devastating, especially for children. Statistics show 1 of 5 who get it die.
And even for those who survive, half are left with serious long-term neurological problems. It's brutal.
Absolutely. And the core reason it's so severe is that this parasite has figured out a way to get past, probably the most secure defense we have, the blood-brain barrier.
Right, the BBB.
And for years, understanding exactly how it breaches that barrier has been a major roadblock. It's limited our ability to develop better treatments.
Exactly. Which brings us to today's deep dive. We've got a fascinating new study that seems to have finally pinpointed the mechanism of attack.
Yes, using a really advanced bioengineered model of the barrier.
So, our mission today is to explore this research, figure out that exact moment the brain's defenses crumble, and talk about a drug, actually an already approved drug that researchers found might help patch up the damage.
Which is a critical shift, right? Moving beyond just killing the parasite to actually protecting the patient's brain during the infection.
Yeah, it's about managing the host response.
Precisely. And this is so important because, frankly, older lab models just couldn't capture the full picture of this neurovascular damage. They were too simple.
So this new approach could really pave the way for what scientists are calling adjunctive host-targeted treatments.
Medications that help the human body, specifically the brain in this case, withstand the assault while other drugs are working on clearing the actual infection.
Okay, so before we get to the new model, let's quickly recap the basic problem in cerebral malaria. It's essentially a neurovascular complication.
Right. It starts with the P. falciparum parasite reproducing inside red blood cells.
And then those infected red blood cells, they start to accumulate, right? They sort of stick inside the brain's tiniest blood vessels.
Exactly. That's called sequestration. And that accumulation physically disrupts the vessels, causing inflammation and restricting blood flow.
But the really catastrophic part, the point of no return almost, is when the blood-brain barrier itself fails.
That's when you get the fatal brain swelling. It's called vasogenic edema. Essentially, the brain starts to flood because the barrier is compromised.
One of the researchers, Livia Piatti, used a really helpful analogy. She described the BBB like a network of tightly sealed pipes designed to prevent leaks.
Okay.
And she said the malaria parasite is somehow able to create cracks in those pipes, causing leaks of, infected fluid into the brain tissue.
That cracked pipes analogy is quite vivid, isn't it? It really captures the idea of a structural failure in the brain's defenses.
It does. But how do you actually watch those pipes crack in a controlled lab setting? That's been the challenge.
And that's where this major leap in bioengineering comes in.
The new 3D blood-brain barrier on a chip, you mentioned it earlier. It sounds like the most complete lab model of this barrier we've seen for infection studies.
It really is. What makes it different is, its complexity and its architecture. Older models were often just flat 2D layers of cells.
Right, like cells in a petri dish.
Exactly. Which doesn't really mimic the true environment. This 3D model, though, it includes fluid flow, like blood pressure, and crucially, it incorporates the 3 key cell types that actually build and maintain the barrier in our bodies.
Okay, so what are those 3 types?
You've got the endothelial cells, which form the actual lining of the blood vessels.
The pipe walls themselves.
Yes. Then you have supporting cells called parasites, which wrap around the vessels.
Okay.
And finally, the star-shaped astrocytes, which are brain cells that connect to the vessels and play a huge regulatory role.
So why is having all 3, especially the astrocytes and parasites, in that 3D setup so important for making the barrier work properly in the model?
It's all about the communication.
Yeah.
In the body, the astrocytes and parasites provide essential structural support, but they also send signals to the endothelial cells.
Telling them to do what?
Telling them to form those incredibly tight connections, those seals that make the BBB so effective. Without that complex 3D interaction and signaling, You just don't get a barrier that functions realistically enough to study properly.
Got it. So they built this sophisticated multi-component pipe system. Then, to figure out how the parasite breaks it, they didn't just look at the initial clogging, the sequestration. They focused on something else.
That's right. They zeroed in on what they called the parasite's most explosive stage.
Which is?
The moment of egress. That's when the parasite, having multiplied inside a red blood cell, bursts out quite violently, actually to infect new cells.
Ah, okay, so it's the breakout moment.
Exactly. And that breakout releases a flood of parasite-derived molecules and debris right into the immediate environment of the blood vessel.
So they hypothesized that this moment might be the trigger for the barrier failure. How did they test that?
They used fluorescent tracer molecules, basically tiny glowing particles they added to the blood side of their 3D model.
Okay.
If the barrier is intact, those tracers shouldn't cross into the brain side. But if it's leaky...
They'll see the fluorescence show up on the other side.
Precisely. And they compared what happened under normal conditions versus when the barrier is exposed to either infected red blood cells or, crucially, just the concentrated fluid collected during that parasite egress stage.
And what did the tracers show?
A really dramatic increase in leakiness. When they added the egress products, the passage of those tracer molecules shot up 6.5-fold.
Wow, so that confirms the pipes were definitely cracking open right around the time the parasites burst out.
Absolutely. And the visual evidence from microscopy backed it up strongly.
What did they see under the microscope?
They used confocal and electron microscopy, which give incredibly detailed images. They actually saw the formation of large gaps between the endothelial cells, literal holes appearing in the vessel lining.
Actual physical damage.
Yes. And significantly, these gaps were often found right near where parasite egress was happening most intensely. The damage was localized to the breakout zones.
Okay, so physical gaps appear, but was it just tearing or something else going on at the molecular level? You mentioned the cells sealing themselves.
Right, the junction proteins. They act like molecular rivets or glue, holding the endothelial cells tightly together.
Yeah.
What the researchers found was that the cells exposed to egress products actually started producing less of these crucial sealing proteins.
So the cells were actively dismantling their own seals?
In effect, yes. They saw a downregulation, a decrease in the production of key tight junction genes, specifically mentioning Claudin-5 and Z01,
Okay.
and also an adherence junction gene called VE-cadherin. So you have the physical disruption near the egress points, plus the cells are being signaled to weaken their own connections globally.
That's a critical link. The physical damage coinciding with this sort of molecular self-sabotage, was there an underlying biological pathway driving both?
There was. They found evidence of a widespread inflammatory response kicking off in all three cell types: in the model, the endothelial cells, the parasites, and the astrocytes.
So the whole neighborhood gets inflamed.
Pretty much. And the key signaling pathway that was strongly activated or upregulated across the board was the JAK-STAT pathway.
JAK-STAT. I know that's often involved in inflammation and immune response.
Exactly. It seems the products released during parasite egress act as a potent trigger for this JAK-STAT signaling.
And that signaling cascade then leads to the inflammation.
Which in turn seems to instruct the endothelial cells to downregulate those critical junction proteins, making the barrier leaky.
Wow. So the parasite isn't just like passively clogging things up. It releases stuff during egress that hijacks the host's own inflammatory system.
And turns it against the brain's defenses, weaponizing the host response, essentially.
Okay, if the host's inflammatory response, driven by JAK-STAT, is a major part of the problem, what does that immediately suggest as a potential treatment strategy?
Well, it suggests that if you could block that specific JAK-STAT signal, you might be able to prevent the barrier from becoming leaky, even while the parasite is present.
Protect the pipes by calming the inflammation.
Precisely. And that led them directly to test an existing drug, a known inhibitor of that pathway.
And that drug is Ruxolitinib.
That's the one. Ruxolitinib is already clinically approved for other conditions, and it specifically inhibits JAK1 and JAK2, key components of that pathway.
So a perfect candidate to test in their model. What happened when they added Ruxolitinib along with those damaging egress products?
The results were quite striking. Ruxolitinib effectively prevented the massive increase in barrier permeability caused by the egress material.
It stopped the leaks.
It did. Functionally, it acted exactly like that patch for the cracked pipes we talked about. When they looked at the molecular level, they saw that the crucial junction protein Z01 stayed properly located at the cell borders, maintaining the seal.
So it preserved the structural integrity.
Yes. by apparently mitigating that whole destructive inflammatory cascade triggered by the parasite products.
How effective was it quantitatively? You said permeability went up 6.5-fold with Egress alone.
With Ruxolitinib added, they measured an almost 24-fold reduction in permeability compared to the Egress condition without the drug.
24-fold reduction. That's huge.
It's a massive stabilization effect, yeah. It essentially kept the vascular barrier intact during what seems to be the most damaging phase of the parasite's interaction with the brain vasculature.
And the really significant part here is that ruxolitinib isn't some experimental compound.
No, exactly. It's an approved drug. And interestingly, it's actually already being investigated in clinical trials for uncomplicated malaria, mainly looking at its effects on the body's overall immune response.
So there's already a clinical pathway opening up.
Right. But this study adds a crucial new dimension. It suggests ruxolitinib might not just help manage systemic inflammation, but could directly protect the integrity of the blood-brain barrier in severe cerebral complications.
So it becomes a true adjunctive host-targeted therapy. You treat the infection with antimalarials, and you potentially treat the deadly brain complication with ruxolitinib simultaneously.
That's the potential, yes. It could fundamentally change how we approach treating the most severe forms of malaria, focusing on preserving brain function while clearing the parasite.
Now, this 3D BBB on a chip sounds incredibly powerful, but like any model, it must have limitations. What did the researchers themselves point out?
They were quite clear about that. First, the complexity itself is a bit of a double-edged sword. Building and running these sophisticated microfluidic chip models requires specialized labs and expertise.
So, not easily scalable for all research right now.
Not yet, perhaps. Another point is that while the barrier function is much better than older 2D models, the actual permeability rates measured in the chip are still a bit lower, meaning slightly tighter than what's estimated for the real human BBB in vivo.
Okay, still not a perfect match physiologically.
Not perfect, but much closer. And probably the biggest limitation they mentioned is the cellular complexity.
Right. You said it had the 3 main cell types: endothelial, parasites, astrocytes. But the brain environment is obviously much richer than that.
Exactly. The real neurovascular unit involves immune cells, neurons nearby, microglia, which are the brain's own immune cells.
So what's the next step to make the model even more realistic?
Well, the group leader, Maria Bernabeu, stated that their next move is precisely to start incorporating those other cell types.
Adding immune cells, microglia, maybe even neurons.
Yes, to build an even more complex and hopefully more physiologically accurate model of the human brain's vascular interface, making it as close to the real thing as possible in the lab.
Okay, so stepping back then. The big picture here seems to be the critical moment of danger in cerebral malaria looks like it's that explosive parasite egress.
Releasing factors that trigger this intense local inflammation,
which then activates the JAK-STAT pathway in the surrounding cells,
causing them to effectively dismantle the blood-brain barrier seals,
leading to those devastating leaks and swelling. But, remarkably, an existing drug, ruxolitinib, shows real promise in blocking that specific pathway and patching the leak.
It really highlights the power of these advanced bioengineered models, doesn't it? Being able to recreate such a complex biological barrier in vitro allowed them to dissect the mechanism and, importantly, rapidly identify a potential therapeutic, a repurposable drug, no less.
Yeah, targeting the host response, not just the bug itself. That offers a speed that you just don't get with traditional drug discovery pipelines sometimes.
Absolutely.
So the success here with this 3D BBB on a chip, pinpointing a therapy for something as complex and devastating as cerebral malaria, it really makes you wonder, and this is maybe a final thought for you, the listener. If this kind of advanced bioengineering can tackle the challenge of getting past, or in this case, protecting one of the body's most fortified barriers for malaria, what other diseases could be next? Think about neurodegenerative conditions like Alzheimer's or Parkinson's, where the BBB is also implicated, or even the difficulties of delivering drugs like chemotherapy into the brain.
Could this BBB on a chip approach unlock treatments for those conditions too?
Exactly. It feels like a glimpse into a future where key parts of drug discovery and understanding disease might happen not just in animals or patients, but growing on a microchip in the lab.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.